This is the second half of Chapter 2, focusing on chemistry of life.
First, there is a field of science called biochemistry, which is the life of chemistry. So, how chemistry is related to life and why it is essential for life. So, in biochemistry, we study the molecules that compose living organisms, which are carbohydrates, fats, proteins, and nucleic acids. And we understand why we need these things and how they help us understand cellular structures, basic physiology, nutrition, and overall health. Chemical elements are the simplest form of matter to possess unique chemical properties. They are a pure substance, one single type of atom. Each element is identified by its atomic number, which is the number of protons in the nucleus. So every element is numbered based on how many protons it has. It doesn't matter how many electrons or neutrons it has, just protons. And it has to have that many protons. If it has a different number of protons, it is no longer that element. So the protons give it its identity and its number. The periodic table is arranged in order, starting at 1 and going up. So the first element on the table is going to be hydrogen, which has 1 proton. And then the next one would be helium, which has 2. And then there'd be lithium, which has 3, and so forth. They go in order by protons or atomic number. Every element has a 1 or 2 letter symbol. There are 24 elements that are important in our body out of the 91 natural elements. 6 of them are very, very abundant in our body, and they're very essential. We really need these. These 6 are oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. They are very, very abundant, and they make up about 98% of our body weight altogether. Trace elements. These are tiny little elements that there's very little trace of in our body, but they are also important. We also have minerals or inorganic elements that we get from soil, plants, food that we eat, things like that. These make up 4% of our body weight, and this mainly consists of calcium and phosphorus. Body structures such as bones and teeth really rely on things like calcium. Enzyme function relies on very important minerals. Nerves and muscle functions also rely on some of these minerals and elements. Okay, let's talk about atoms and what they are made of. So an atom has a nucleus, which is the very center. A nucleus is composed of protons and neutrons. So a proton is a single positively charged unit. It has a mass of 1. So a proton is positive and it weighs 1 AMU or atomic mass unit. Neutrons. Neutrons are also in the nucleus and they have no charge. They're called neutrons because they are neutral. They also have a mass of 1. So if we were trying to figure out the atomic mass of something, we would add up how many protons and neutrons that element has, and that would give us the atomic mass, because each proton and each neutron are worth 1 atomic mass unit. So if we had an element that had 5 protons and 5 neutrons, that element would have a mass of 10. Electrons. So we also have electrons in atoms, but they are not in the nucleus. They are outside of the nucleus, orbiting around, like the planets do around the sun. They are orbiting around in electron orbitals. Okay, they never go into the nucleus. They always stay outside. Electrons virtually weigh nothing, so they do not contribute to the atomic mass. We don't need to count them to add up weight at all. And they are negatively charged. So on the outermost shell of an atom, so atoms can have anywhere from 1 to 7 shells for the electrons to float around and orbit in. The outermost shell, no matter which number it is, the last one, has electrons in it that are called valence electrons. So these are electrons in the outermost shell. These electrons are important for bonding. Okay, an atom is typically, as an atom sits on the periodic table, it's going to be electrically neutral. So it's not going to have a positive charge or a negative charge, but it's going to have positive and negative subatomic particles. So they're all going to have protons and electrons, but it's not going to have a charge. And that happens because all atoms that are on the periodic table are neutral because they have the same amount of protons as electrons. So if they have 10 protons, they have 10 positively charged particles. But they're also going to have 10 electrons, which are going to cancel out the positive charge of the protons. So essentially, the charge would come out to 0 because one would cancel out the other. So every element on the table, the way it sits, is neutral. There is no charge. We can have charges, but on the periodic table, they're all neutral. So let's talk about isotopes. Isotopes are variations of atoms that have the same amount of protons and the same amount of electrons, but a different number of neutrons. So if you remember back to when we talked about protons, protons give an atom its identity. So a hydrogen atom is always going to have one proton. If it has more, it is not hydrogen. It has to have one proton, very specifically to be a hydrogen atom. It can have a different number of neutrons and a different number of electrons, but it has to have one proton. So when we change the number of neutrons, or we find an atom that has a different number of neutrons than it should have, it becomes an isotope. So it's still the same element, it's just a different version of it. So it's like different colored cars. They're all the same type of car, they just have different colors, right? So we have hydrogen, which has one proton, no neutrons. Then we can have hydrogen 2, okay, which is also called deuterium. It's hydrogen 2. It has one proton and one neutron. So it's still hydrogen, but it has an extra neutron. So it's a different version. It's an isotope. So the extra neutrons add to the atomic weight. Because remember, neutrons counted for one atomic mass unit. So if we add one neutron, we have to add one atomic mass unit. So isotopes of an element are typically very, very similar to the regular element because they're still the same element. They still have a lot of the same properties, but the weight is going to be different. So when we talk about them, we need to note the weight of them because that's how we figure out what is in these isotopes. That's how we can identify if it has a different number of neutrons. So hydrogen 2 would be hydrogen with a mass of 2. We would subtract one proton from the number of 2 because we know it has one proton, and then we would know that it has one neutron as well. Along with isotopes comes radioactivity. So this is where we get radiation from, except for the sun. So isotopes of an element typically behave the same as the element because, again, they are still the same element. That hydrogen 2 was still hydrogen. But their physical behaviors can be slightly different. So radioisotopes are usually unstable. So these isotopes will decay and give off radiation, which is called radioactivity. Every element has at least one radioisotope. So every element has one version of instability where it is radioactive. We can use intense radiation to do things like destroy molecules. We can use this for cancer treatment, or a body can have this happening naturally, which can create free radicals, which cause cancer or mutations. So we can make it happen scientifically, or we can have it happen naturally. The physical half-life of a radioisotope is the time that it takes for 50% of that isotope to decay. That's a half-life. The time it takes for half of the life to go away. And then the biological half-life is the time required for 50% of it to leave the body. So when we use radioactive isotopes, we really use them to date fossils and things that we find that are really, really old. When something dies, it has carbon in it, and specifically carbon-14 is the radioisotope of carbon. It decays. So we can test by how much carbon-14 is still in a fossil, how long it has been decaying, or how long it has been dead. So we use these in the body for dating things that are really old. We use them for x-rays, we use them for cancer treatment, and then they happen in the body on their own. We can encourage them to happen by, doing things that are not healthy, smoking, exposing ourselves to toxic chemicals. Those things can cause the isotopes to become less stable, and it can cause things like cancer or genetic mutations over time. So we talked about isotopes being an atom that has a different number of neutrons than it has on the periodic table. So the periodic table is the normal neutral version of every atom, every element. Isotope is a version that has a different number of neutrons. An ion is the version that has a different number of electrons. So now remember, electrons are not neutral like neutrons, and they don't weigh anything. So they're not going to change the weight of our atom, but they will change the charge because electrons are negatively charged. So an ion is a charged atom, an atom that has a charge of either positive or negative. This happens when electrons are transferred from one atom to the other, and this happens during bonding. An anion is a negatively charged atom, and a cation is a positively charged atom. I was taught, like way back when, to remember them: angry ants and happy cats. Anion is negative, cation is positive. So ions that have opposite charges are going to attract, because opposites always attract, right? Magnets, the negative and the positive end are going to attract. Same thing is going to happen in ions. A negative ion is going to want to attract a positive ion because of the magnetivity. So ions, again, they form through bonding and chemical reactions. So if we have a neutral atom that has 6 protons and 6 electrons, if we lose 1 electron, now we have 6 protons and 5 electrons. The higher numbered subatomic particle is going to win in the charge. So if I have 6 positives and 5 negatives, I have more of the 6, which is more of the positives. So it's going to become positively charged by the difference. So if there's 1 different, it's going to be a +1 charge. If there were 2 extra protons than electrons, it's going to be a +2 charge. If we lost 3 electrons and we had 6 protons, now we have 3 more positive than negative. It would be a 3 plus charge. And remember, we can only give away or lose electrons, never protons, because if we change the number of protons, we change the atom entirely to a different element. So protons can be given away or gained. If we gain an electron in an atom, now it has more negative. So if you have 6 protons and you gain 2 electrons, you have 8. We have 2 extra negatively charged things, making it a 2 negative charge. Okay, so ions are charged particles. They are very, very important in controlling some of the pumps in our body for our heart to work, our muscles to work. So ions do play a huge role in anatomy. Chemical bonds. So atoms want to bond with other atoms to satisfy something called the octet rule. And if you think about octet, oct means 8, right? So the octet rule means that every atom wants 8 valence electrons. If you remember back when we talked about electrons, a valence electron is the electron in the outermost orbital. This orbital is important for bonding. That's its whole job, is to bond with other elements and other atoms. So if you look at this picture, we have this atom. It has 1 electron in its outer shell. It's not happy. It wants 8. The ultimate goal is 8. Okay? If you have 8, you complete the outer shell. The most stable form of an atom comes with 8 valence electrons. So every atom's goal is to get 8. They can get these extra electrons by bonding with another atom. So atoms who have 8 valence electrons on their own, they are referred to as the noble gases. They don't want to bond because they're satisfied all on their own. They're not reactive. They don't bond. They just kind of hang out and do their own thing because they're happy. So the octet rule, 8 valence electrons is the ultimate goal for every atom. If they don't have 8, they will bond with other elements to get 8. And if they do have 8, they don't bond. Once they bond with another element to get 8, they are satisfied, and they usually stay bonded until there's some kind of chemical reaction that pulls them apart. Although every atom without 8 valence electrons wants to bond, they don't do it in the same way. So there are different types of bonds, and they depend on what kind of atoms are trying to make these bonds. So first we're going to talk about covalent bonds. If you think about the prefix co, it means like working together, cooperating. You know, there's co-parenting, there's co-workers, cohabitation. That all means together, right? So in a covalent bond, atoms share electrons. You have two atoms that are missing some of their valence electrons. Maybe they have 6, maybe they have 7, but they want that 8th one. And if they work together and they share what they have, they can both satisfy the octet rule by getting 8. So when things covalently bond, they both end up with 8 valence electrons. They don't give away or take, but they share. If you are bonding and sharing a single set of electrons, it's a single bond. If you're sharing a double set, or 4 in total electrons, it's a double bond. And if you're sharing a triple set, or 6 electrons, it's a triple covalent bond. Usually covalent bonds occur between two nonmetals. If you look at this picture, element A, whatever that is, it doesn't show us the nucleus, so I have no idea what it is. It has 7 valence electrons. So does atom B, 7 valence electrons. They both need one more to satisfy the rule of 8. So if they share the single pair that they each have and share that 2, now they each have 8 because they have 6 of their own, but they're sharing 2. So this is how a covalent bond works. They are sharing and they are working together. Here's an example of a covalent bond, but if you look in the middle where the two atoms are overlapping, there are 4 electrons in there. So this is not just a covalent bond, it is a double covalent bond because they are sharing 2 pairs of electrons. And if you count the electrons on the outer shell, including those 4, each atom of oxygen has 8. Each one has 4 of its own, and then the 4 shared. Another example of a covalent bond are these nitrogen atoms. So these nitrogen atoms are sharing 3 sets of electrons with each other. This is a triple covalent bond, and if you count, the nitrogens each have 2 electrons, and they are sharing 6, which makes 8 for each. Again, they have to satisfy that octet rule of having 8, and they are sharing what they lack, basically. The second type of bond is an ionic bond. If you remember what an ion is, it's an atom with a charge. So an ionic bond is when 2 charged atoms bond together. So in an ionic bond, there's no sharing. One atom is going to give its electrons to the other, and that atom is going to take them. So you're going to have an atom that lacks electrons and an atom that has too many valence electrons. So the way it works is if you are closer to 8, so if you have 5, 6, 7 valence electrons, you want to take, because it's easier to get to 8 from 5. And then if you have less than 5, so if you have 1, 2, or 3, you're going to give away your electrons. It's easier to get down to 0 and default to that next orbital shell down. So when we're looking at electrons, they have orbital shells, and some of them have 1, 2, 3, 4, whatever it is. The outermost one is the most important. And it doesn't matter what number that outermost shell is. It could be the first shell, the second shell, the third shell. It doesn't matter, as long as it's full. So if an atom has 1 valence electron in its outermost shell, it can give that 1 away, drop the outer shell because now it's empty, and default to the next level down. And the way it works when you're filling shells, you have to fill a shell completely before you can move on to the next one. So if an atom has 4 shells and it has 1 valence electron in the 4th shell, shells 1, 2, and 3 have to be full before they can move on to the next one. So if they drop that outer shell's 1 electron and default down 1 shell, that shell is full. So it's easier to get rid of the 1 electron if you have 1 valence electron than to go find 7 more. So typically, if they have 3, 2, or 1, they will give them away. And if they have 5, 6, or 7, they will take. So a metal is usually the ones that have 1, 2, or 3 that are going to give them away. And a non-metal are the ones that have 5, 6, or 7 that will take. So when we have ionic bonds. We have metals and nonmetals bonding. The metals will give the electrons to the nonmetal to satisfy them. So let's say we have a metal that has one valence electron. It's going to look to bond with a nonmetal that has 7, because that nonmetal only wants 1, because it doesn't want more than 8, it just wants 8. So the nonmetal is going to take 1 electron and only 1 electron. And then that nonmetal, or that metal, wants to get rid of that 1 electron. So if that metal gets rid of the 1, it defaults to the next shell. It's happy and full. If it gives that 1 away to the nonmetal that is missing it, it's happy and full. We have an ionic bond. So when we have ionic bonds, things will only bond if it satisfies the octet rule. You won't have something bond if it doesn't complete itself. So in that situation, sometimes 3 different things can bond together. So if you look at this picture down here, we have a magnesium in the middle and 2 chlorine atoms. So chlorine has 7 valence electrons. It's a halogen. How many valence electrons does it want if it has 7? Well, it only needs 1 more to be happy, right? So a chlorine ion is only going to take 1 valence electron. It will never take more, because it will put it over its 8, and it will put it in the next shell and give it too many. It doesn't want that. It just wants 1. When it gets 1, it's happy. It's done bonding. It's over. So we have this magnesium ion that has 2 electrons to get rid of. It has 2 valence electrons in that outer shell that it wants to drop, so it can default down to the other shell. So this magnesium atom comes around and it finds a chlorine atom and it says, hey, let's bond. I'm going to give you 2 electrons. The chlorine says, no, I don't want 2. I only want 1. I only need 1. I only want 1. But I have my friend over here, this other chlorine, that will take the other 1 because he also wants 1. So this magnesium atom is actually going to bond to 2 chlorine atoms, because now the chlorines are happy and the magnesium got rid of both of its extra electrons in the outer shell. So sometimes it's not a 1-to-1 ratio. Sometimes it's 2-to-4. Sometimes it's 3-to-1. Sometimes it's 5-to-1. It just depends on what the atoms are. But an ionic bond, they're going to give away or transfer electrons from one atom to the other in order to satisfy the octet rule of everything involved in the bond. This magnesium ion would not bond with 1 chlorine. If there weren't 2 chlorines available, it wouldn't try to bond at all. So it has to be enough for it to satisfy itself. Magnesium would just float around still being magnesium with 2 valence electrons, looking for something better to bond to. So any chemical reactions that happen in our body are referred to as metabolism. We have different types of reactions that happen in our body. We have ones that release energy, which are decomposition reactions. They break covalent bonds and they make smaller molecules. So when we are making things smaller, we're obviously breaking them apart, right? So we're breaking the bonds in those things and we're making them smaller into smaller molecules. This is called catabolism. Then we have where we are storing energy. We're building things up. We're making new bonds. And every time a bond is made, energy is stored within that bond. So if we're building things up and making new bonds, we are storing energy. This is called anabolism. So in our body, we have metabolism. And within the metabolism, there are 2 different types, catabolism and anabolism. One is breaking things down, and one is building things up. Here's a diagram of cell metabolism. So if you look, it has food. Food goes in, it becomes macromolecules. So when we eat food, it's not just eating chicken, it's protein, right? So it becomes a micromolecule, which is it's a macromolecule in our body. So we have our proteins, our carbs, our starches, whatever we ate, right? They come in and they get broken down by anabolism, right? Organic chemistry. So organic chemistry is nothing other than the study of carbon. It is everything about carbon and why carbon is so important and how many things carbon can do. Carbon makes pretty much all of our macromolecules in our body. So carbohydrates, lipids, proteins, and nucleic acids are all made out of carbon. So they are all part of organic chemistry. So carbon. Carbon is a neutral atom that has 4 valence electrons. So it doesn't fall into the category of having 3 or less and wanting to give away, or having 5 or more and wanting to gain valence electrons. It falls into its own category. It has 4. Carbon likes to bond with other carbon. Carbon easily bonds with itself to create backbones of carbon. These backbones of carbon can be used to form many things. They can be used to form starches, lipids, proteins, all these things, just from the backbone of carbon. Once a backbone is formed, those carbons will start attaching to things like hydrogen, oxygen, nitrogen, sulfur, and other elements in our body very easily. So when these things cluster together and they attach to the carbon backbone, they create functional groups in our bodies that are used for different things. These functional groups also make many of the properties of the macromolecules, lipids, proteins, nucleic acids, and carbs. So all of those things are created with carbon backbones and functional groups, which is the carbon backbone bonded to other things like oxygen, hydrogen, and nitrogen. Macromolecules. I was just talking about them, but they're very large organic molecules that are formed by carbon backbones. They are polymers, and examples of these macromolecules, again, are carbs, lipids, proteins, and nucleic acids. All these things are formed by carbon. I mentioned the word polymers. So what is a polymer? What exactly does a polymer do, or does it come from? So a polymer is a macromolecule that is made up of a repetitive series of similar subunits. So if you look at this picture, the polymers are on the bottom, and it's 2 white, 1 black, 2 white, 1 black, 2 white, 1 black. It's repetitive, right? It's a pattern. That is a polymer. It's a subunit of something that attaches repetitively to make a chain. Those repetitive subunits are monomers. They are single subunits that can bond to each other to make a longer chain, such as a polymer. And the act of attaching to make a polymer is polymerization. So remember, the macromolecules are all polymers. So macromolecules, carbs, lipids, proteins, and nucleic acids, are all these chains of monomers that are attached together. Carbohydrates are hydrophilic organic molecules. And remember, hydrophilic means water-loving. So these are things that can be broken down easily by water. We especially break them down easily to make ATP, which is energy. These are our most efficient source of energy. Sugars, starches, breads, potatoes, bananas, anything like that is going to be where we get our energy from. When naming carbohydrates, they come from the root saccharine. So anything that has saccharine in it is a carbohydrate. Like monosaccharides, disaccharides, those are all carbohydrates. And often in our body, carbohydrates are paired with either fats, which are lipids, or proteins. When they are paired with them, they become glycolipids or glycoproteins. Okay, monosaccharides. So the prefix mono means one, and saccharide, we know, means carbohydrate. So monosaccharides are a single version of a carbohydrate. They are the simplest, smallest version. They are made up of monomers, so a single molecule. And some examples are glucose, galactose, and fructose. Because these are so simple, it's very easy for our bodies to break them down and use them for energy. We can break them down really quickly without a lot of extra work. They're really easy for our bodies to dissolve and just use for whatever their purpose is. Next, we have disaccharides. So the prefix di means two. So a disaccharide is two monosaccharides bonded together. And when they bond covalently because they're made of nonmetals. Remember, nonmetals bond covalently. So some examples of disaccharides are lactose, which is milk sugar. It's glucose plus galactose, or maltose or sucrose. Those are all examples of two monosaccharides together to form a disaccharide. And now we have polysaccharides. So the prefix poly means many. So a polysaccharide is a long chain of many, many monosaccharides. And in order to be considered a polysaccharide, there has to be at least 50 monosaccharides in that chain. Some examples of polysaccharides are glycogen, starches, and cellulose. Glycogen is how we store energy in our liver. There's some in our muscles, our brain. Starch is energy storage in plants that we can eat and digest. And then cellulose is structural molecules in plants that is important for our daily fiber intake, but we do not digest cellulose. Moving on to the next macromolecule, we have lipids. Lipids are hydrophobic organic molecules because they are fats. So if you guys think about when we put fats or oils in water, they separate, they don't dissolve. It's because they're hydrophobic. They don't like water. So fats have more calories per gram of actual weight. So if you have the same amount of fat and carbs, there are more calories in the actual fat, but we cannot break them down as efficiently as we do for carbs. So we get way less energy out of the fat. Usually we cannot break them all the way down. We end up storing them, which is why if you eat a lot of fats, you usually gain weight. We just store them. We don't break them down and use them. There are 5 primary types of fats in the human body, and not all of them are bad. Some of them we really need. These are fatty acids, triglycerides, phospholipids, eicosanoids, and steroids. The first type of fat we're going to talk about, or the first type of lipid, is fatty acids. So fatty acids are essential for our bodies, and we have to get them from food. We don't make them on our own. Fatty acids can either be saturated or unsaturated. So saturated fatty acids are saturated with hydrogen. So if you look at this picture here of this carbon backbone, remember we talked about carbon makes up all of these macromolecules. So if you look at the middle here, this whole backbone's all carbon, 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 okay? And then if you look, every single carbon can make 4 bonds. And if you look at them, they are all full with hydrogen. So there's not any open carbon. Each carbon has 4 bonds. Okay, 2 of the bonds are going to be to other carbons because it has to connect to the next carbon to make a chain. However, every other bond available on these carbons is taken up by a hydrogen. The beginning has a hydrogen and the end has a hydrogen. This thing is full. Okay, there are no double bonds between the carbons because everyone is having a single bond with a hydrogen. An unsaturated fatty acid means that it is not fully saturated with hydrogen. There are some areas where the carbon backbone has double bonds between the carbon, meaning that if we broke this double bond right here on this bottom picture, we could actually make that a single bond between the 2 carbons, and you could attach a hydrogen to both of those carbons. So there's room to add more hydrogens because carbon is double bonding to itself instead of to other hydrogens. So unsaturated just means that we can add more hydrogens to make it more saturated. All right, triglycerides. These are neutral fats. These are usually a glycerol with 3 fatty acids attached. If you look at this picture, there are 3 of the blue thing. Those are your fatty acids. And then there's this reddish colored thing over here. That's your triglycerol, or your glycerol and your triglyceride when you put them together. The primary function of these is energy storage, and they also help with insulation and shock absorption. If you think about what fats do, fats are squishy and they're thick, right? So they're going to absorb shock. If you have a lot of fat on your body and you fall, you're going to have more padding. Babies have a lot of fat. They have a lot of padding. Helps them with shock absorption, and it also helps them with temperature regulation because their brains are not mature enough to regulate the temperature completely on their own. That extra fat on their body helps them accomplish these goals. Dietary oils are triglycerides, and when we think about these oils, they are usually liquid at room temperature. So olive oil, vegetable oil, those things are liquid at room temperature, but fats are solid at room temperature, which is fats from like steak. If you cut fat off a steak, it's going to be a solid. It's not going to be liquid. So you can get your triglycerides from your oils and fats. And just remember the oils are liquid and fats are solid at room temperature. All right, phospholipids. These are really important because these make up our cell's membranes. Every cell in our body has a membrane, and it's made with a phospholipid bilayer. We're going to talk a lot about phospholipids next week when we talk about the cell membrane. But for now, you need to know that a phospholipid is very similar to the neutral fats, except for instead of just being fatty acid, there's a fat and a phosphate group together. So it's a phospholipid because it's a phosphate and a fat. Okay, these, like I said, these are your structural foundation for your cell membrane. And they're amphipathic, meaning that they have a hydrophobic and a hydrophilic portion. So if you think about what this is made of, there's a phosphate and a lipid. One side has to like water and one side has to not like water. Which thing doesn't like water? Well, it's probably going to be the fatty end, right? Because we know that fat is hydrophobic. So when we look at these phospholipids, it has a hydrophilic head, which is the phosphate group, and it has a hydrophobic tail, which is the fat part. So they are a two-part fat. One part is polar and one part is nonpolar, and they're essential to our cells functioning. So here we have eicosanoids. These are 20 carbon compounds. They're pretty complex, but they are very, very similar to hormones because they can send signals between cells. Some examples of these are prostaglandins, which they act very much like hormones, even though they're not. But they have really big roles in our body in things like blood clotting, producing hormones. They're really important for helping labor progress. So these are eicosanoids. Steroids. So our body has steroids in them naturally. They are a fat with 17 carbon atoms attached to them in some sort of shape of a ring. So steroids are synthesized from other parent steroids. So you have one steroid that makes other steroids, because remember, synthesize means to create. Steroids are really important for nervous function and for things like sexual reproduction, growth, stuff like that. 15% of our cholesterol comes from the diet, and 85% of it we make ourselves. And this cholesterol is what our bodies use to make the steroids. So some examples of these steroids are cortisol. This is a steroid that we release when we get stressed. Progesterone and estrogens. These are hormones released during ovulation or labor. So those are the female reproductive hormones. There's testosterone, which is the male reproductive hormone, and it's also responsible for facial hair, the deep voice, the more square structured of a man's face, and then bile acids. And bile is the thing that is in our stomach that helps us break down food. So these are all steroids, and they are also a lipid. Okay, cholesterol. So you always hear people saying, oh, I have high cholesterol, or I need to lower my cholesterol, like it's something that's bad. Cholesterol isn't necessarily bad. We do need it, but you can have too much of the LDL, which is the kind that contributes to these problems that people talk about when they have high cholesterol. So HDL is a high-density lipoprotein. So it's a high-density fat protein combined. This is cholesterol that is good for you. It helps you prevent cardiovascular disease and helps your heart function properly. It keeps your arteries clear, things like that. LDL is low-density lipoprotein, and this is the bad cholesterol. This is the cholesterol that clogs up your arteries. It's very, very sticky. It gets in your arteries, sticks to the wall, and sticks to itself, eventually causing cardiovascular issues. So make sure you know that HDL is good. I remember this because HDL is high. You want the high to be high. LDL is the low. You want the low to be low. Proteins. Proteins are the building blocks of our body. They build everything. So proteins are made up of a polymer of amino acids. So amino acids are the building blocks of proteins, which are the building blocks of our bodies. When the amino acids connect to make a polymer, if two or more are attached to each other, it's called a peptide. Okay, and the way they attach with each other is called a peptide bond. So two or more amino acids makes a peptide. When two or more amino acids bond together to make that peptide, that bond is a peptide bond. Okay, when lots and lots and lots of those bonded peptides come together, they make that polymer, which makes our proteins. So proteins are named for the number of amino acids that they contained. Dipeptides have 2, tri have 3. Oligopeptides have anywhere from 3 to 15, and poly means 15 to 50, and then proteins are 50 or more. So a peptide chain of at least 50 amino acids makes a protein. Okay, and proteins are very, very complex, very twisted and intricately bent and turned molecules, so they can have lots and lots of amino acids. Let's talk about the structure of proteins. So proteins look like a big jumbled up mess, but there is a very specific way that they have to be twisted and folded to work. So if you look at this picture, this looks like purple swirly ribbon that's all clumped together. There is a very specific reason for that protein to be folded the way it is. When a protein is folded correctly, it's called conformation. Okay, so this is the protein. It receives the code to fold this way and this way and this way because of the job it's going to do. Every protein is folded and bent for the job that it needs to perform. When it does it correctly, it's called conformation. They work and they're good to go. If they do not fold correctly or they unfold, it's called denaturation. So this destroys the protein's ability to perform its job because it's not bent, folded, or twisted correctly. So these can happen in production, or it can be damaged from something else like heat or an illness in the body, a change in pH, anything like that. So when a protein becomes denatured, it doesn't mean it's trash. We can always send it back to get refolded, and then it can be checked again to see if it's functional, and if so, it can move on. If it's still not folded correctly, we can send it back again. So just because it unfolds doesn't mean it's trash, and just because it folded correctly doesn't mean it's always going to stay correct. It can always get ruined, and then we can always fix it. Our body has lots of checks, so it can go through these processes to make sure that the proteins we are making are folded correctly for the job that they're intended to do. Since proteins make everything in our body, here are some examples of what they actually more specifically make. So proteins are responsible for structures, specifically keratin and collagen. This is bones, cartilage, teeth, hair, skin, nails. These are all things that need protein to actually build and grow. So if you're really, really lacking protein in your diet, you might struggle to grow your hair, or you might struggle to, have nice skin or grow your nails nicely because you're lacking in that. Communication, neurotransmitters such as hormones and other signalers, like the receptors that bind to receive the signals from the rest of the body, rely on proteins. Membrane transport. Cell membranes have proteins in the cell protein channel receptors in the cell that accept proteins to move things in and out of cells. Without proteins, this would not happen. Proteins can act like catalysts. So a lot of enzymes that are used to speed up reactions rely on proteins. Recognition and protection. So some proteins are really important for creating white blood cells and helping our immune system to make antibodies and other things like that. Movement. The biggest thing that most people know that proteins do is it's responsible for building and repairing muscle. A lot of people that spend a lot of time in the gym or doing sports are going to be eating increased amounts of protein because it helps repair that muscle after you've worked it very hard at the gym or in sports or whatever kind of exercise you're doing. So it's really important for our muscles. And then it also helps cells bind together. So it's important for cell adhesion. So I mentioned that proteins can be catalysts with enzymes. So enzymes are proteins that function as catalysts. And a catalyst is something that speeds up the rate of reaction. So if we have a catalyst present or an enzyme present, it can make the energy needed to make that reaction happen a lot less. So these make things happen in our body that would take a long time to do without the enzyme. So if you're lacking enzymes, your reactions are going to slow down. A substrate is the thing that the enzyme actually acts upon. So whatever the enzyme is coming to act upon is the substrate. And then enzymes are usually named substrate plus ACE. So if the enzyme is working on the amylose substrate, it's going to be called amylase. Okay, so this is the action that enzymes take. So the first thing an enzyme does when it's catalyzing something is the substrate binds to that enzyme. When that binding occurs, it makes an enzyme substrate complex. So whatever the enzyme is trying to speed up the process of, it's going to come attached to the enzyme, and then it's going to make a enzyme substrate complex. Okay, the enzyme is always unchanged in this process. So the enzyme is never affected by catalyzing things. it can do this over and over and over and over again without anything happening to itself. So enzymes are never consumed by the reactions, and they can catalyze millions and millions of reactions per minute. Okay? Other things that can speed up the reactions or change the shape and function of an enzyme are temperature, pH, and other factors such as bacteria and things like that in the body. They can alter the enzyme's ability to complete its job by altering its ability to bind to a substrate. So if it cannot bind to the substrate, it's not going to be able to form that complex and do its job. Enzymes need certain pHs to properly work. Different enzymes have different pHs because if you think about in our body, there's going to be spots that have higher pHs and lower pHs. So one spot in our body that's going to have a lower pH is our stomach. Remember, when we eat food, our stomach releases acids. So our stomach is going to be very acidic compared to like the inside of our mouth. Okay, our saliva is not acidic like our stomach acid. So an enzyme that works in the stomach is going to have to be able to function at a way lower pH than an enzyme that works in our mouth. And then the optimum temperature for our bodies is 37 degrees Celsius or 98.6 degrees Fahrenheit. That's perfect body temperature. Enzymes function best at that temperature. If our bodies are warmer or colder, they're going to slow down in how fast they can catalyze reactions. Enzyme cofactors. So a cofactor is a non-protein helper molecule whose presence is needed for enzymes to do their job. So this cofactor is like the coworker of the enzyme. It needs it. Okay? About 2/3 of our enzymes require a cofactor. Cofactors can be a whole bunch of different things. They can be iron, magnesium, calcium, and these work by binding to the enzyme and triggering a change that activates the actual site on the enzyme that does the work. So without the cofactors binding to the enzyme, the active site doesn't get turned on. The enzyme cannot do its job. And then there are coenzymes. So coenzymes are things that are working with enzymes, such as vitamins. They do the same thing as the cofactors. The enzyme relies on them to get its job done. All right, metabolic pathways. A metabolic pathway is a chain of reactions that are catalyzed by enzymes within our bodies. Metabolic pathways can be turned on or off, and these can be achieved either by binding or by feedback. So if we need something made in our body, a feedback mechanism is going to turn on saying, hey, we need to make more of this. And then we'll make more. Once we reach the optimum amount, another feedback mechanism is going to send a signal back that says, hey, we have enough, stop making this. And then the production will stop. When we need that thing again, the mechanism will turn back on and the process will continue. So metabolic pathways are controlled by enzymes, and they can be controlled usually through feedback mechanisms. All right, so the next macromolecule is a nucleotide. A nucleotide is an organic compound with 3 very specific components: a nitrogenous base, a sugar, and at least 1 phosphate group. Okay, so some examples of nucleotides are ATP, or DNA. ATP is adenosine triphosphate, and it is energy for our bodies. This is what energy comes from. We have to make these molecules to make energy. We break them for energy. And then DNA. DNA is our genetic code. Nucleotides make up the building blocks of DNA. So nucleotides are very, very important for our body. 1, because it writes our genetic code, and 2, because if we don't have them, we don't have any energy. So here is ATP. Okay, so this is an adenine, a ribose, and 3 phosphates. Its name is ATP, adenosine triphosphate, and the triphosphate means 3 phosphates. So ATP is the energy that our body uses. Energy is stored in the bonds between the phosphate groups. So if you look at this picture, it shows the little lightning bolt. The energy is stored in those bonds. And when we talked way back about chemical bonds, when things are bonding together, they're storing energy in the bonds. When things are breaking apart or unbonding, they are releasing energy from those bonds. So in order to get energy out of ATP, we have to break a bond. The bond that is broken is the bond between the 2nd and 3rd phosphate group, because that's the one with the most energy. So we break this bond, we use the energy that comes out of it, and then we have ADP. ADP is not usable for our bodies. We can only use ATP. But remember, when we're getting our energy out of ATP, it's the second and third phosphate group that has the broken bond. That is the only bond that we break to get energy. We don't break this bond between the first and second, only the second and third. So once that bond is broken, we have ADP, adenosine diphosphate, as in two phosphates. Like I said before, we cannot use ADP. It doesn't do anything for us, but we don't throw it away. We can take that ADP and put another phosphate group on it to make it ATP again, because it's all it's lacking is that third phosphate. So we can get that phosphate through food. We eat food, we get phosphates. Those phosphates go find that ADP molecule, and they reattach to make it ATP again. Once it is ATP, we can break the bond again and use it for energy. And the cycle will continue and continue and continue. We will always reuse the ADP until there's something else wrong with it. If it's damaged in another way, we cannot reuse it. But it's always reusable as long as it is ADP. We can add the phosphate, break it again, get our energy. And lastly, we have nucleic acids. Nucleic acids are polymers of nucleotides. So we talked about nucleotides and ATP. If we put a whole bunch of these nucleotides together, we make nucleic acids. Nucleic acids make up DNA and RNA. DNA is deoxyribonucleic acid. Okay? This makes up the genetic code for everything in our body. Okay? It writes the instruction for everything in our body, how it should work, how often it should work, what it should do. That's DNA's job. It also makes up RNA. So RNA is responsible for carrying out genetic instruction for making other things or making new things, and make sure that amino acids are producing proteins correctly, and that when our cells divide and split, that it's done correctly. So these things are very important, and we'll talk about DNA a lot more when we get to chapter 4 and we talk about the genetic code.